So the first thing that we're going to do today is we're going to do a little drawing. And this is going to be just totally rudimentary, but we're going to draw a leaf. So here's a leaf. On the outside of the leaf, you've got a layer of cells. We're going to say that this is like the top of the leaf up here, and this is the bottom of the leaf. So we're looking at like a little cross section, okay? So, the parts of the leaf that I want you guys to be familiar with, just for our discussions, are going to be the next layer down. Okay, there's a big layer of cells down here. Okay, and this layer is called the palisade mesophyll. This layer here is called the palisade mesophyll. All right. Then there's going to be these layers, or these types of cells that are called xylem and phloem, and that makes up the vascular tissue. So if you've ever looked at the plant and seen like the little veins on the leaves, right? Those are vascular tissue. That's where stuff is actually going to be transported. You should remember some of that from bio honors, right? And so you've got these bundles of vascular tissue that look like this. These are the veins of the leaf. Okay, and I've drawn them in black here because, interestingly enough, when we talk about xylem, which is the one that transports water from the roots to the leaves, it's dead at maturity. Those are dead cells, and they only work when they're dead. Okay, so this is going to be vascular bundle. Okay, now there's going to be a layer of cells around the vascular bundle like this. And if you wanted to come up with a neat name for them, you would come up with something like their actual name, which is the bundle sheath cells. So if you know what a sheath is, like if you're a knight or something like that, you would put your sword into a sheath, like that's the little you know what I'm talking about? Makes that sound. It's like sometimes made out of leather, sometimes made out of metal, right? Sheath. It's the thing that surrounds the sword. So these are the bundle sheath cells because they're the things that are surrounding the vascular bundles, right? So these cells are bundle sheath cells. All right. And then the last thing that I want you to know about are these little tiny mouth cells. Specifically, there are two cells, and there are two cells that are called guard cells. Okay, and these guard cells, they make up these little things that look like mouths, and the mouths, they're called stomata, which translated means tiny mouth. Okay, so why are the stomata there? The stomata are there because the plant needs to exchange oxygen, and the plant is water type and air type. And so if it can't exchange oxygen to release its oxygen and take in carbon dioxide, then it's going to die. Okay, so it opens up these tiny mouths just like you open your mouth and you breathe out carbon dioxide and breathe in oxygen. The plant does the opposite. The plant breathes out oxygen and breathes in carbon dioxide, right? Okay, so let's talk about the problems that photosynthesis has. And we're going to refer back to this drawing a bunch. So, the biggest problem with photosynthesis is that the key enzyme for the Calvin cycle, which is rubisco, it has a problem, okay? And its problem is that it doesn't really care what it binds to as long as it's binding to something, right? So we say that rubisco has an equal affinity for CO2 and O2. Okay? That means that rubisco will bind almost equally as well to O2 as it does to CO2. All right? And that's a problem because we know that rubisco's job, its name is ribulose bisphosphate carboxylase, and if it binds to O2, instead of adding a carboxyl group, it's going to add an O2 group. And that's an issue, right? Everybody agree that that's a big problem? And once it adds the O2 group, then the whole process of the Calvin cycle breaks down because then you don't get the production of PGA and everything just stops, right? So we can look at this objectively and say, wow, really good job, evolution. You really messed this up. Why didn't you evolve something better than this? If rubisco is like one of the most common enzymes on the planet, why is it so crappy? That's the question, right? And the answer to that is that when rubisco evolved, there was no oxygen. Rubisco evolved to make photosynthetic things, right? Well, before there were photosynthetic things on Earth, there was no oxygen. The only reason we have oxygen on Earth is because of photosynthetic things, right? So early Earth, when rubisco evolved, had little to no atmospheric oxygen. Okay, so to bring some current events into this, we just found water on Mars, flowing water on Mars, right? That's pretty neat. And we know water has O2 in it, right? It has oxygen in it. It's H2O, right? And we also know that Mars, if you've ever heard Mars called the red planet, have you heard that before? Yes? Does anybody know why it's red? Oxidized iron, right? So it's got oxygen for sure, right? It's rust, all right? Question is, though, where is that oxygen coming from? Is it in the atmosphere? Is it if we landed there, could we just go and breathe in a bunch of oxygen? No, it's all gone. It's in the rust. It's in the water. It's bonded to other things. So there's no free oxygen in that atmosphere. Earth was the same way. There was no free oxygen around. When there was free oxygen, it went through an oxidation reaction and turned into something else, right? If just overnight something happened and every photosynthesizing organism died, we would not live here very long, right? And the reason for that is that the O2 would not just get used up by us, it would get used up by all kinds of things, right? By rusting, by fires, right? All these things take O2 and they turn it into CO2. And then the only thing that turns it back is photosynthesis, right? Plants are pretty important, right? But before there were plants, there was no O2. So that means that when rubisco was evolving, there was no reason for it to be able to exclude O2 because there was no O2 there. It wasn't until the plant started to get really good at photosynthesizing that now they had a problem because they were becoming too good and the waste product, O2, was starting to bind to that enzyme rubisco. Make sense? So we can say that if O2 binds instead of CO2, the RUBP molecule is wasted and must be sent to the cytoplasm to be broken down. So that's a lot of extra energy that this thing is exerting now. Not only does it create the wrong molecule, so that's losing some energy there, But then it has to actively transport it out into the cytoplasm. Then it's got to be broken down by a hydrolytic enzyme, and then it's got to be transported back in. That's a big waste. Evolutionarily speaking, that's super bad. All right. This process is called photorespiration. And photorespiration is why my yard can never be the way that I want my yard to be. Okay? So I'm from Ohio, and overall, I don't like Ohio very much. It's not my favorite place in the world. It's kind of horrible, actually. But one of the best parts about Ohio, maybe the best part, is that all of the grass in Ohio is Kentucky bluegrass, right? Or the vast majority of it, at least. Okay? I don't know if you know about Kentucky bluegrass, but it is awesome, right? It's thin, it's soft, right? It's got this awesome, like, bluish green tint to it. It's the best grass there is, okay? So, if I really, really, really wanted to, I could take a bunch of Kentucky bluegrass sod and drive it down, right? And I could sod my lawn in it come, like, maybe, I don't know, late October, right? And it would last in my lawn until probably June, right? And then it would all die. And the reason for that is that it's so hot in Florida that it would begin to lose all its water, and when it loses all its water, it panics, and it closes its tiny mouths, stomata, so it doesn't lose any more water out of it, right? And then it starts to build up the waste products of photosynthesis inside. The waste products of photosynthesis are O2, right? And then it doesn't have any more CO2 because it's using it up, right? And so then instead of going through photosynthesis, it's only going through photorespiration, and it will die as a result of that, right? So I have to go with this grass that is, you know, Bahia or St. Augustine, which, spoiler alert, in Ohio, we call that crabgrass, and we kill it, like, immediately. It's like, this is horrible, it's a weed, get it out of here, right? So why does this other grass grow, this other terrible grass? Why is that thing that always grows down here? Well, it's because it's going through a different type of photosynthesis, right? There's 3 types of photosynthesis, and it's going through an evolved type, an adapted type for this Florida weather, okay? 3 types of photosynthesis. Whoa. Type 1. We won't talk too much about it because you guys know about it already. It's called C3 photosynthesis. And this is the normal kind. In other words, the kind that we've been learning about the whole time is C3 photosynthesis. Okay? It's got the light reactions, it's got the Calvin cycle, uses Rubisco. Okay? This was the first to evolve and most common. Most common and first to evolve. Okay? It creates a 3-carbon chain after CO2 fixation. What's the 3-carbon chain called that's created immediately after CO2 fixation?
3PG.
Yes, 3PGA. Okay, and that's why it's called C3 photosynthesis, because it's creating a 3-carbon chain. C3, 3-carbon chain, right? It is prone to photorespiration. So anytime that you have a very hot environment or a very dry environment, C3 photosynthesis is not going to be the way to go, right? Because it's going to have to go through too much photorespiration, because it's going to keep its stomata closed too much. All right? Questions? All right. Next, C4 photosynthesis. Like our friend the St. Augustine grass, right? That's what's happening there is C4 photosynthesis. And C4 photosynthesis we can think of as a physiological adaptation to this problem that it has, okay? Physiological, anatomical, structural, they all essentially mean the same thing in this case, and that is that it's going to just change which part of the plant that photosynthesis takes place in. Okay, so anatomical adaptation for hot environments. Okay. This is your tape. Hi. Sure, no problem. Okay. So, what is this adaptation then? Well, all it's going to do is it's going to change where it does photosynthesis. The photosynthesis is going to occur only in those bundle sheath cells, right? So, Calvin cycle only occurs in bundle sheath cells. Okay? All the other cells in the plant still have a job though. Their job is carbon fixation. Okay? So all other cells use the enzyme PEP carboxylase to create a 4-carbon chain called malate via carbon fixation. So a couple things you need to know about this particular thing we just wrote there. First of all, 4-carbon chain called malate, right? That 4-carbon chain is where C4 photosynthesis gets its name from, because the initial step of carbon fixation makes malate, right? Which is a 4-carbon chain, so they call it C4. The second thing that you need to know is that it uses this thing called PEP carboxylase. PEP carboxylase is an enzyme that captures CO2 very similarly to the way Rubisco does, except for it doesn't ever bind to O2. It selectively binds to CO2 and not O2. Okay? Make sense so far? All right. So then the last step is that I guess I'll indent here, that the malate is transported to the bundle sheath cells to allow for normal Calvin cycle reactions. Yeah.
Do they still make like a 6-carbon chain later on, or is it just?
Yep. So it actually goes through a normal Calvin cycle. The only thing that happens is the malate then gets broken down into CO2 inside the bundle sheath cells, and then the bundle sheath cells can immediately use that CO2, and then it sends the 3-carbon chain back out to be turned back into malate. Okay. Last one is called CAM photosynthesis. CAM photosynthesis. Okay? And CAM photosynthesis is the most evolved of all of them. It's the most sort of hardcore here. And so this is more of like a behavioral adaptation. So a behavioral adaptation to prevent photorespiration in hot, dry environments. And the term that you're going to see used to describe hot, dry environments is the term arid. Okay, if it says arid, it means hot and dry. Okay, so the key to CAM photosynthesis is that this behavior that they have is that they will only ever open their stomata at night, and they keep their stomata closed all day. Okay, so stomata stay open at night, and are always closed during the day. The type of plants that go through CAM photosynthesis are a group of plants that we refer to as succulents, right? So succulents are things like aloe vera, right? It's kind of got a little like chunkiness to it, right? Cacti are succulents, right? And so their idea, or their adaptation, is that they open their stomata at night and they allow all this CO2 in, and they keep making sort of a storage molecule for the CO2. The storage molecule is called malic acid. Okay, so at night, CO2 is fixed to produce malic acid. And the malic acid is where it gets its name from. CAM actually stands for Creslucian acid metabolism, right? Which you can just think of as acid metabolism because it's using malic acid, right? And that malic acid that's being made during the day is going to be stored in the central vacuole. Okay, so to produce malic acid, which gets stored in the central vacuole. Okay, and then at night, or sorry, during the daytime then, the stomata close, and then whenever the plant needs to increase the concentration of CO2, it just takes some of that malic acid out of the central vacuole, and it breaks it down into CO2, and then there's plenty of CO2 there, right? So during the day, awesome, thank you. Malic acid is broken into CO2 and used for the Calvin cycle. Okay, so in CAM photosynthesis, all of the cells are going through the Calvin cycle, just like before, but the difference is they only do it during the daytime, and then at night they spend all of their energy, or I don't want to say they only do it during the daytime. Most of their energy at night is used to make this malic acid stuff, and then that is used, the malic acid is used to be able to continue the Calvin cycle during the day. The question, though, that I was going to ask, based on what I just said, was why are we less likely to see the Calvin cycle happening at night? Because we're less likely to see it happening at night. What? What? Really?
Is it because there's less ATP and NADPH being made because there's no light?
Yeah, there's no light, so there's no ATP and NADPH to go into the Calvin cycle. So we're not saying that none of it happens at night, but once you run out of ATP and NADPH, you can't have that go on anymore. So most of the energy is used to make malic acid at night. I had forgotten, just slipped my mind somehow. I was going to bring in, for you guys, there's a plant that we grow out here all over the place, the purple stuff. You guys seen the little purple plants? Those are called purple queen. And there's another type that is in the front area there called wandering Jew that's purple and green. And it's got awesome stomata. And I'm going to bring it in on Monday, and I'll pluck one off and I'll keep it in water so the stomata stay open. And then we can take it out, and you can watch under the microscope. You can watch the stomata closing. Yeah, we'll do it Monday. Questions? I think I told you guys that the reading guide was due today. Or no, I didn't tell you that. I had figured it out already.
Monday.
It's due on Monday, right?
Yeah.
Yeah. And then on Monday I'll get you the new one.